Good morning. Can you hear me in the back? Yeah, great. So my name is Karel Svoboda. I'm the director of the Allen Institute for Neural Dynamics. We're one of the sponsors of the Odin meeting together with the Dandy group at MIT. And also we received generous support from ASAP, the group, the Aligning Science Across Parkinson's. We're really also interested in open science and neurophysiology. So the organizers are Saskia de Vries, Nima Dehghani, Satra Ghosh, and David Fang. And thanks to them for doing a great job on really short notice, right? And so I just want to say a few words to set the somewhat idiosyncratic kind of few words perhaps to set the stage. So what is open science, right? We probably all agree open science is sharing methods and code in well-documented code in common repositories for collaboration, transparency, and doing science together. In addition, of course, to data in widely adopted, ideally useful formats, right? And this is sort of often summarized in the FAIR principles. So the data should be findable, they should be accessible, they should be interoperable and reusable. And these terms really refer to metadata, the data that describes the experiment. So the primary data is understandable in a machine-readable fashion. And I think in a young field like systems neuroscience, right, a lot of the discussions this week will be about systems neuroscience. The data is extremely complex and changing. We're really falling significantly short of the FAIR principle. There's one reason I think we need to have meetings like this, like the Odin meeting. So why should we engage in open science, right? Why do open science? And I think it's perhaps there hasn't been a better time for this kind of a symposium than now. One reason is the American research enterprise is really under assault, right, and with uncertain outcome. And I'm sure that's on the minds of many of you, even if you're from abroad, right? And one way I think to brace ourselves and survive the onslaught is to do better intentional collaboration, and open science is really one way to go about that. Now, related perhaps, open science is also about transparency and reproducibility, which builds trust in science. And so trust in science is an important topic beyond the halls of science. It's really quite very much in the ether. And open science principles that encourage good stats, good modeling, right, good data science really help with building trust in science. Now, neuroscience, as all of you know, and this is really a topic today, right, is remarkably interdisciplinary, right? I mean, many of the high-impact studies nowadays involve multiple collaborators, maybe across multiple labs, and open science allows you to do more efficient collaborations with a larger number, especially delocalized collaborators. And something that we've learned a lot at the Allen Institute is that open science, we've learned it over and over again, that open science is really increases the return on investment for your hard-won data, right? Because any neuroscience study, the kinds of stuff that many of you do in your laboratories, really when we publish papers, we only scratch the surface of the data set, and pretty much any high-value study should be made available, or the data and materials should be made available for additional kind of discoveries. Now, finally, and I think this is also a theme of the meeting, I think quite important, is that what is the ultimate goal of open science and open data is to create resources that allow multi-scale modeling of brain function, right? However these models look, whether or not they're foundation model or biophysics-inspired models, there are many variants of that, but we will have to learn how to collate insights, right, about neurodynamics across multiple studies done in different labs. And I think good data habits, good data science, good open science really is the only way to do this. So this year's Odin meeting focuses on neurophysiology and the future of neurophysiology, and especially on the multimodal, multi-scale aspects of neurophysiology. Neurophysiology is about spying on neurons and the dynamic processes of neurons using mainly electrophysiological or optical methods. Neurophysiology measures the dynamics of neurons that underlie neural computation and behavior, but as such, I think that we all agree that neurodynamics has to be interpreted in the context of not only the behavior produced by the animal, but also ultimately the neural circuit that the neuron belongs to. And so we want to integrate the recordings across scales. I have a couple cartoons to illustrate this. Is a kind of experiment that many folks are doing, not just in rodents, but in primates, perhaps multimodal analysis across multiple brain regions in the context of behaving animal. Ideally, these recordings are anatomy guided, right, so that we can record from brain regions that interact with each other. We can glean a bit of mechanism how one brain region interacts with another. So the metadata, the data science challenge here is to describe the recorded neuron's location, because that tells you where in the brain it is and how it might interact with other parts of the brain. And of course, the animal and its behavior. And in addition, great advances are being made in enumerating the brain's cell types, right? And the cell types are, of course, where evolution acts on the brain, right? Evolution kind of modulates gene expression, which ultimately specifies the connectivity rules in the brain. And so cell types are kind of the nodes of circuit diagrams. And we have to kind of interpret and learn to interpret neural dynamics in the context of these evolved neural circuits. And this ultimately requires us that we track the neuron type. And this is becoming more and more possible. And I think for next generation neuron models, brain models, we have to really take structured neural circuits and cell type into account. So localization of neuron here is not enough, because each brain region contains each little brain volume, even tens of micrometers, will harbor multiple cell types that we need to identify to see how the recorded cell type fits into the circuit. And we'll hear a lot about this. This matters really for neural computation, for interpreting the neurodynamics that we record with neurophysiology. So I'll give you just one example from Drosophila. We'll hear a couple of examples, and we would not do badly by looking at what is happening now in Drosophila research in terms of multimodal, multi-scale analysis of neural function, because there are a bunch of resources that come into play, like, for example, the connectome and tools that allow one to access specific cell types in the brain that allow one to do this multimodal analysis of neural circuits and computation in the intact brain in the context of behavior. This is just one example. Looming selective neuron in the lobular plate of the fly, right? To interpret the looming response to looming stimulus, you have to sort of know where the neuron is, because where the neuron is, that tells the fly how to move to avoid the fly swatter, right, or not. And that's sort of the location matters, but also the cell type matters, because the cell types tells you about the wiring of the neuron, how these different dendrites move into regions of the lobular plate. This is all in the visual system of flies, where different kind of motion directions are computed, which already tells you just from the structure of the circuit how the looming stimulus is computed. So linking, again, recordings here and location to neural circuits gives you insight here into neural computation at the level of individual neurons. And this kind of specificity is likely also the rule in mammalian circuits. We shouldn't kid ourselves just because we don't know a lot of this level of specificity in mammalian circuits that it's not there. I'll just give you one very brief example from ongoing work at the Island Institute for Neurodynamics. This norepinephrine neurons in locus coeruleus is a tiny neuronal population in the mammalian brain that projects widely across the brain, has functions in many behaviors, including anxiety-like behaviors, sleep, wake cycles, has roles in learning, and these neurons are also among the first to degenerate in neurodegenerative disorders. And what we found is that these neurons are diverse. They're not a homogeneous class. They come in different projection classes. For example, locus coeruleus neurons that project from the pons here to the cortex, or from the pons to the spinal cord, and very little overlap, right? And then recordings from these different types suggest also that they have different, again, computational roles in the brain. This is in a task when an animal has to learn trial by trial to make choices about the availability of reward in particular places, and the neurons that project to the front of the brain show these kinds of reward prediction errors that you may know from dopamine neurons, performance signals that are poised to drive learning, so bigger responses when the reward was bigger than expected, smaller responses when the reward was smaller than expected, but the neurons that project to the back of the brain are excited by lack of the reward. They might set up the system to change behavioral strategies. So again, neurons projecting to the front of the brain are excited by a reward prediction error, the neurons that project back to the brain by lack of reward. So in this tiny neuron population, different projection types have different functions. They also have different, actually, biophysical properties, different spike shapes. They're in slightly different regions of the LC. They have different gene expression patterns, and really we need to bring all of this stuff together to understand neural computation and ultimately how these neurons generate their sort of specific response types and how they affect downstream neurons. So here are some key themes that I'm going to look for over the next couple of days, and I'm sure there are a lot of stuff we can add to. What are the key obstacles to collaboration on shared data, and how do we remove them, especially the kind of complex multimodal, multi-scale data that we're talking about. Should we think about what kind of multimodal data might be required? We should think about kind of collating across different studies for modeling the brain. Of course, different kinds of data will be required for models that just are about dynamics of neurons that could be non-mechanistic or mechanistic models. How do we share that data, aggregate that data across studies for the purpose of modeling? How should multimodal data be structured? This is really at the kind of data science level and data standardization. How do we share data physically? What kind of platforms do we use? I feel that the cloud has to play an important role. We can't just move data from data center to data center. The size of the data doesn't scale, but the uptake of cloud in the kind of research and academic community is still very low. So how do we do this actually practically? How do we minimize barrier to entry and reduce friction? And this is also related. How do we reduce data movement, cost of use, licensing, and so on and so forth? And I hope that we discuss all of these topics in the context of exciting open science. I have no doubt that we'll hear a lot of exciting open science over the next couple of days. Okay, so I'm not going to say anything else and just fire up another a couple introductory slides for Nima, who will go through the schedule with you. Okay, Nima.
All right, thank you all for being here, and thank you, Karel, for the introduction. So just something quick to mention is that I want to thank Karel, and because when it was 2 years ago, we talked about perhaps bringing Odin to Allen Institute. He graciously agreed, and then later we put together this program along with Saskia, David, and Satra. So I've been given a sort of a script here that I'm supposed to read out for you. So, and I'm not the best at that, because when I was in elementary school, I got fired from theater for not being able to follow the script. So there are a few things actually I would like to mention before we go forward. One is that basically each of you has a badge. Make sure you have your badge all the time. And if you want to go inside the building, you're welcome to hang out in the atrium area. But if you want to go to see your friends or colleagues or someone in the other floors, please call them and ask them to synchronize with the security, because you're not supposed to wander around. So that's one thing. And on the back of your badge, you have a QR code that has all the information about the program. And it also has two colors. Basically, these are for panel sessions. I got the black ones. It's like which tells you which room you're supposed to go to. We will go over the panel discussions and parallel sessions later today, but just to give you a highlight, the idea of having this Odin put together initially with Satra at MIT was that we are entering an era that we have thousands of recording channels, we have massive multimodal data coming to the surface, and this, whether it's like you're doing electrophysiology with like optical imaging or any sort of these things, you have to deal with all this sort of data, and making sense of this and managing it is extremely hard and challenging. And as a community, we need to come together and we need to find solutions for this. And this is not a job that a given lab can do only, and we need to work on this together. And especially, as also Karel mentioned, given the current climate, it is important to recognize that we're all in this together and we could do better together. So wherever we are. So is the case about modeling and analytics. So the software that are being created, the tools that are being provided, if they are released publicly, they will help other colleagues to do better. They help us to do sort of like analysis that is verifiable. That is super important, right? We want to enter a new age where things are not going to be done only by one lab and they're not replicable, and then there's this like single study that showed something. We want to go beyond that. We want to go beyond that level. We want to integrate information across the scales of observation, the different type of experiments we want to do. And to do that, we need to create data standards and also have mathematical foundations, computational foundations that are shared by the community and verified consistently. So, and there's also NIH data management and sharing policy. What was left at NIH, we don't know, but the idea is that if something is publicly funded, eventually it should be publicly available. And this is good for many reasons, because there's time and money, and there's also reducing of animal sufferings necessary. And when a given discovery happens in a given lab, you want to be able to go back to the same data set later. You want to go back and see what was done. And if a postdoc or a student left the lab, you don't want this to get lost in this, in some hard drive that no one knows what was done. And without all this, we can say that we do need new horizons for basically having to deal with this sort of multi-scale data sets. So we have 3 days. The first day is going to be about foundations and infrastructures. The second day, knowledge extraction and AI integration. And the third day is going to be neuroinformatics and future integrations. On the first day, we'll have Peter, who I'll let the podium to be handled by him in a minute. So Peter comes from Copenhagen, and he studied at University of Copenhagen before moving to US to do his postdoc with Yuri Brzezicki, and he works on memory, and he does have very good contributions to both softwares and infrastructure for devices for open science. And after that, we'll have a session 1, which is about advanced neurophysiological devices, then followed by a talk by David Kleinfeld, who will give us the challenge talk, and then we'll have the session 2 about integration, and we'll go to parallel breakouts. So during parallel breakouts, remember that each of you is supposed to be in some parallel breakouts, and we will be escorted up to the specific rooms. I don't know where they are in this building, but someone will help us, and we'll walk there, and then after that, we'll have the summary of that and panel discussions. And tomorrow, we'll have Jacob Make giving the keynote, and we'll have 2 sessions from Rade that are actual insights and building and benchmarking AI models, again followed by parallel breakouts and panel discussion. Sorry, it says network reception is tomorrow, but network reception is today, not tomorrow. And the third day, Saskia will give us the third keynote, and followed by preparing data for multi-scale data and analysis, and computational neuroscience and multi-scale modeling, and a panel discussion. There is no parallel breakout sessions on that day, and then we'll have a talk by Satra about the future of Odin. And last, I wanted to thank you all for being here, for making the journey, especially for our European colleagues who came here, even if they were given the warning, some of them, that, if you go to US, be careful. But, hopefully we'll pass this. We have this hashtag, and we want to thank the funders, and also I want to thank Kendall Slovak and Senem, as well as the AV, who helped us with organizing this. Without it, it wouldn't be possible. So I ask Peter to join us, and let us have a good discussion. And also remember, we want this to be engaging. So please participate in the discussions, in the panel discussions, in the parallel sessions. We want to hear what are your thoughts. It's not a one-way